Members can all be seated. We can start with the second session. James Hickman obtained his PhD in biochemistry and chemistry from MIT. He has 6 patents and 60 publications, including 9 book chapters. He's a professor of chemistry, biomolecular science, electrical engineering, and computer science, as well as the founding director and professor of the Nanoscience Technology Center at the University of Central Florida. Before doing that, Dr. Hickman held the position of Hunter Endowed Chair in the Engineering Department at Clemson University. Dr. Hickman, Professor Hickman. So we have 20 minutes.
Yes, sir. All right. So thank you much for the invitation. This is a very interesting meeting. I've had lots of interesting discussions and also enjoyed listening to discussions from in the audience. So today I'm going to talk about some neuroscience. I'm going to, and also how nanoscience has affected that. So we do neuronal tissue engineering in the hybrid systems lab at UCF. I'm not going to talk about the in vivo applications today, but we do a lot of work in neuromuscular junction formation, and most of the other people who do tissue engineering of organs are eventually going to have to worry about this. Okay, and so we do a lot of work in that area. We also look at prosthetic interfaces and CNS and PNS repair. What I'm going to talk to mostly today is in vitro applications and trying to create functional systems. Most systems for drug discovery, for trying to use as disease models, pretty much are single-cell assays, or if you got to go into animals and eventually human beings. What we want to try and do is create functional systems between single cells and animals and humans. We're looking at organs. A lot of other people looking at organs as well. We have a lung project. But we're mostly focused on our lab as control systems, which is motor control mimics, look at the spinal reflex arc, memory formation. We have a lot of projects looking at Alzheimer's disease and memory deficits, as well as Peter showed you some work on cardiac pacemaking. The big nanoscience aspect of this is controlling the interface between the biological and non-biological components. So we can pretty much take any cell type and integrate it with a non-biological material using surface chemistry. And I'm going to show you lots of examples of that today. So back in 1995, we published the first paper where we applied engineering principles to cell culture, okay, where we basically took 5 main variables and found that if we controlled those 5 main variables, okay, that I could get anybody in my lab to do cell culture reproducibly. Okay, I could take engineers, I could take chemists and physicists, and they all do cell culture because we can control these 5 variables. We use self-assembled monolayers as the surface. The nice thing about those, we can make geometric patterns, so we can actually then control circuits and make circuits in dishes. Media formulation, so we're working mostly with neuronal systems. We want to try to approximate CSF, which is no serum. Cell age, we're now pretty proficient, probably the only group in the world that can do not only embryonic cells, but adult cells from rat, mouse, and human, neuronal systems from both spinal cord, hippocampus, and cortex. Cell preparation early on was an extremely important variable, and now it's pretty much standardized. And we can also do different cell types, all the different cells of the central nervous system, as well as other cells. We can also look at immune cells and start looking at some immune diseases like this MS and things like that, autoimmune diseases, and how that affects the nervous system. All right, so this is a very, very busy slide. I could probably sit there and talk about the aspects of this slide for over an hour, but I won't. But pretty much what you do is you can take an adult rat, you take the spinal cord out, and then you do a series of preps where you're looking at density gradients, and that allows us to separate different bands of cells and allows us to get a fairly pure population. One can also go back in and do a selective antibody panning for specific cells and get a much more pure population if we wanted to. We also use self-assembled monolayers. This is our primary growth surface, which is a triamine functionality, which goes onto the surface using a silane. So we can then put that on top of any surface that has a hydroxyl group. Okay? So anything that has a hydroxyl group, we can put these self-assembled monolayers down on it. We then use glass cover slips or MEMS devices. We then combine it with this media formulation we came up with. And today I'm going to talk about our research where we tried to look at controls that didn't contain Ceria nanoparticles and those controls that contain Ceria nanoparticles. Now I'll talk a little bit about the Ceria nanoparticles next. Okay, so we've already talked about, people have talked extensively about the creation of nanoparticles. I'm not going to go into that so much, other than it's just an emulsion. You can get very nice cerium particles out of that. If they're single crystal, it's a high resolution TEM. You can see the patterns here to show that they're actually crystalline in nature. But you can also see, look at XPS spectroscopy. XPS spectroscopy is a technique where you can look at different oxidation states of materials. And what you can clearly see here is you have a combination of cerium 3+ and cerium 4+. And what we believe happens is that the cerium nanoparticles go back and forth between the 3+ and the 4+ state in a catalytic fashion to scavenge radicals. Now I'm going to try and prove that to you. Okay? But firstly, I want to show you that when we actually take these nanoparticles in an in vitro cell culture environment, Beverly's going to talk to you about, and then talk after this, about what happens when you go in vivo. But in these controlled in vitro experiments, what we found is actually you see a statistical statistical difference in the number of neurons or total cells that are present from an adult spinal cord rat culture with the Ceria nanoparticles and without. And also what you see is over a longer period of time is a lot of cells that would have eventually gone into apoptosis because we're severely injuring these cells when we take them out. Okay? So a lot of them go into programmed cell death that early on you don't see, but what you can clearly see is the neurons survive at a much higher rate in the presence of the Ceria nanoparticles than you don't. So this has, in effect, what we're reproducing is a very ischemic environment. Okay? And so you're controlling that ischemia by putting in a catalytic nanoparticle. A lot of antioxidants have a one-use system. So vitamin E, vitamin C, or something else that has a one-use, takes a radical out of the system, but then it's done. Whereas in these Ceria nanoparticles seem to cycle. That's what we also found is that the cells, you can go in immunocytochemistry, show that we have neuronal cells in there, and also that we can get the recovery of some function, and I'm going to come back to this later on, of the cells, and that they will fire action potentials. In this particular case, single action potentials. So we can take the cells out of an adult rat, acutely dissociate them, injure them, and get them to regenerate at least partial function of their electrophysiological activities. Okay? So how do these systems work? Well, what's happening is the Ceria nanoparticles are cycling between that +3 state and that +4 state. So if you actually take the original solution of the cerium nanoparticles and you hit it with hydrogen peroxide, which is an oxidant, what you do is it changes color. You can actually follow that in the UV-vis spectrum. And over a period of time, it then converts from the +4 back to the +3 state in a serum-free media. Okay? What then, you then hit it with hydrogen peroxide again, and it cycles back. And it cycles back, and you keep doing this over and over again. Okay? Now, the test, though, is can we actually take a cell culture of neurons and hit it with hydrogen peroxide and actually protect the neurons in the hydrogen peroxide culture system? What you see is a very large difference in the survival of the neurons, okay, that have been treated with the hydrogen peroxide in the presence of the cerium nanoparticles. So, in effect, we're protecting them in an in vitro environment from an extreme ischemic event by treating them directly with hydrogen peroxide. Okay, so the cerium oxide nanoparticles are scavenging the free radicals, okay, at a fast enough rate to protect the cells from a very nasty ischemic event. Okay, you can actually see the live dead assay here. All right, so let's say, okay, well that's really cool. You've actually proved that cerium nanoparticles are neuroprotective. And so what we do is we go in, when we're actually extracting the spinal cord out of the adult rats, all the solutions that we put them in have the cerium nanoparticles. They're in every step during the isolation. But what's actually happening during the isolation of these neurons? Because we're the first group in the world that has shown we can actually take adult spinal cord neurons and keep them alive in culture, let alone make them functional again. Okay? So in effect, what we're trying to do is to totally regenerate motor neurons that have been injured. Okay? So what happens during that process? So this is a live dead assay here, which is 6 hours after plating. You can see the cells here, a couple dead cells, and actually see them start to bleb a little bit, put out some processes, start putting out some processes, and you can actually start getting some polarity back in those cells after 48 hours. But if you actually go in, and this, you can't quite see, it's a little bit too bright in here, but we have two different stains here, which is nestin is red and neurofilament is green. So neurofilament is an adult marker, so proving that these aren't a stem cell or precursor cell or something else like that that we're isolating. We're actually isolating out adult neurons from the system. But what happens is you get a co-localization of the nestin, which is an embryonic marker. So nestin is one of the first things that comes up during development when the cells are first going from stem-like or progenitor-like behavior and start going along a differentiating path. So we turn those pathways back on in these injured cells. So in effect, it was mentioned before, we get a de-differentiation of the adult cells to repair themselves. And so you see a co-localization of the adult marker as well as the nestin marker. You see a co-localization, but after about 96 hours in culture, okay, you only see the neurofilament and you no longer see the nestin. So they've gone back in time, okay, in programming, reprogrammed to repair themselves, and now they're actually going back and getting rid of those pathways, and now they're surviving and starting putting out processes. And again, to show that we actually have motor neurons in the system, we've used just about every stain for motor neurons. Islet 1, this is a phase picture here. You can see the islet 1. These cells are both motor neurons. ML1 is another putative marker for motor neurons. Chat, showing that they're positive for acetylcholine esterase. Synaptophysin, they're actually forming synapses. And map to A and B, showing that these are dendritic markers. So we do have motor neurons in culture. Okay? But what happened is that much like in the stem cells where people say they're getting motor neurons, you can do wonderful immunocytochemistry on these systems, but it does not prove that they've actually retained function. Because to be able to get them functional again, they have to find repetitive action potentials. That's what motor neurons do. That's how they actually control the muscles, okay, in your system. So if you want to show that you actually have full recovery of function of the individual motor neurons, you actually have to have repetitive function. And so a very brilliant cell biologist in my group, Mayank Das, came up with this very interesting idea as well, okay, let's try to get these things that are back fully functional. We know there's an activity-dependent innervation that pushes development to its final stages, okay, in development, that actually where the cells have to actually form electrical connections. We said, well, okay, maybe it's not electrical activity, maybe it's the neurotransmitters themselves that is causing the full maturation of these systems. And what he did is he treated this with a combination of neurotransmitters, acetylcholine, serotonin, and glutamate, and actually finished the recovery of these cells back to the same properties they had in vivo, which is now repetitive firing. And to prove that these are actually motor neurons and not interneurons that are repetitive firing, you can go in and see that these are islet 1 stains. It's showing these repetitive firing. These are patch clamped here. And so we can get full functional recovery for the first time. It's been demonstrated in vitro. And I believe it's been indirect evidence in vivo that you get full functional recovery after damage. But this shows that we can actually do this in a very controlled environment. Okay? So, okay, what do we do with this? So we have applications for amyotrophic lateral sclerosis, for motor control deficits. Very interested in ALS because if you get ALS, it's a death sentence. And it's a death sentence for two reasons. It's unlike Alzheimer's where you slowly lose your function over time, okay, but you stop realizing that you've lost it after a period of time. In ALS, you know you have full functional capability until the day you die. Okay? And since in 98% of the cases they don't even know what causes it, okay, you don't have any hope. Okay, so this is, by going and be able to do adult cultures, we're now starting to do adult human cultures. We might be able to start looking at these disease models directly in a controlled in vitro environment, where I can start looking at and asking some questions, okay, from the diseased human cells, okay, in an environment that mimics what occurs in the reflex arc, but in a much more simplified environment. Okay? We can also apply this obviously to spinal cord injury, muscular dystrophy, neuropathic pain, if we can actually make this model. So how would I actually make these systems, okay, in a dish? Okay? So what I can do is I can actually break this up, and Peter Molnar talked a little bit about this earlier. We treat cells as components. What that allows me to do is to create this, to separate that as an engineering problem, where my motor neuron is taking electrical signals to muscle activation, which then does a force or muscle mechanics through the spindle, then reports back from the muscle mechanics, encodes it into electrical potential, and then recreates the reflex arc. The reflex arc is basically the thing when you go in a doctor's office and he taps your knee and your leg kicks. That's your reflex arc. That's what he's testing for. Okay? So by being able to recreate this system, I can look at all these various disease models. Okay? And again, since I can start using adult cells, I can start looking at disease models directly, either from transgenic models or from diseased human tissues. So this is how we try and do it. We try and use surface chemistry. Here we can put a motor neuron, control where its process goes. We can then innervate a myotube that's sitting on an AFM cantilever, so I can then look at muscle dynamics, okay, without having to patch the myotubes. Okay? I can then have a DRG with cycling back through the system and then re-innervating this motor neuron to be able to recreate the reflex arc. What I can tell you right now is we've reproduced every aspect of this circuit. I do not have the time right now to go through all the aspects. What I'm going to do is focus on this one right here, which is motor neuron to myotube, and show that we can actually do this. Okay? And how we do this is we can actually take this self-assembled monolayers and make patterns, use lithographic patterns, and come back in with a second molecule and actually make very nice patterns of various cell types using surface chemistry. Okay? So we can take a pattern such as this, plate out first in myotubes, the very nice myotubes forming where these patterns are here, and very nice myotubes forming patterns here. What you can then do here is coming back in, this is an axon from a motor neuron, coming in, beautiful branching that you would normally see the synapse, okay, going on to a myotube or a muscle, okay, through the systems. We actually get synapses on the pattern myotubes. Okay, and it's actually showing the stain here. This is a myotube. This is an unpatterned system, just showing that we can actually get the motor neurons in a co-culture. This is a serum-free co-culture where we can actually get myotubes or muscle, okay, coexisting in the same media as the motor neurons and also DRG neurons. And again, I don't have the time to show the DRG. All in the same media system that's serum-free. So again, if I want to do drug development, okay, I can't do this in serum because I have lots of factors I don't understand. So I'm doing this in totally controlled environment. So if I want to look at the effect of one drug for, say, looking at muscular dystrophy, I can do that. Or I can look at a drug if I want to try to correct ALS, I can do that as well. Okay? Because I have a very controlled environment. But the various stains here, we can actually see this is a confocal image, different slices in the 3D plane here, showing that we actually have the myosin heavy chain, showing we actually have the muscle cell here, neurofilament, that we actually have adult neurons with their processes coming in here, and also showing that we have neuromuscular junction formation. You'll see these slight blue portions in here. Okay? So, well, okay, fine. You can get myotubes, you can put them on these systems. You said something about integrating myotubes with AFM cantilevers. What are you talking about? So what we want to try to do is put a myotube on top of a cantilever and then use a laser deflection. So when this thing flexes, it changes the orientation of that cantilever, much like atomic force microscopy. That's how it works. We can also use piezoelectric cantilevers. I'm not going to talk about this today. I'm going to talk about this. So we can very nicely, these are the silicon-based cantilevers here. We can grow them out on the surface, and they automatically align along the long axis of the cantilevers. But the nice thing is again, we've also been able to do the interfusal fibers as well as the extrafusal fibers. So we can now start looking at spindle function. At the same time, we're looking at muscle dynamics that are controlled from the motor neuron. And the reason we can look at spindle dynamics is because we can now put, using neuregulin, we can create the interfusal fibers, which can constitute the spindle portion of the reporter unit. Okay? So we can actually have an extrafusal fiber here that's connected to a motor neuron that then stretches, when it bends the cantilever, the interfusal fiber. And that's connected to a dorsal root ganglion sensory neuron that feeds back to the system. So we don't actually have to recreate the entire spindle. We can actually just co-localize two different types of myotubes on the same EMF cantilever. We can then actually have a system where we can pulse generate signals onto these myotubes. This is the laser spot sitting on top of it. You can see the myotube that's running along the long axis of this cantilever. You can run a 1 Hz signal on this and show very nice deflections, very nice control of this system in real time. Showing that we can actually, again, create these systems, okay, such that we can integrate it, biological, non-biological components, to be able to build systems where it doesn't matter which one I'm using as the biological component, which one I'm using as the non-biological component, I can build these systems. I can then build them to try and understand what I need to do once I go in vivo as well. And again, I'm not going to talk about the in vivo work we're doing, but we're also going in vivo. And the nice thing about this, okay, is that because I can create these systems outside the body, I can look at specific effects without having all the complicating factor, okay, that if I go inside an animal or a human being, okay? We say, well, in a lot of cases, you need those complicating factors because you need to understand what's going to happen from metabolic products and things like that. That's true. But what I can do, since I can actually use the same cells I'm taking out of the animal or the human, okay, I can reiterate back and forth between the two. So I can go back and forth between systemic and specific effects. So if I want to look and see what happens with glial cells in the system, I can add this back into the system. We've actually got some very nice myelination models of PNS and CNS axons in the system as well. Okay, so I think I'm probably running out of time. Yes?
45 seconds.
Perfect. So here's my acknowledgments. Like to thank the Hybrid Systems Lab. So Peter Molnar, who already spoke earlier. So I didn't talk about, even though my title has hippocampal, I didn't talk about that because Peter mostly talked about that earlier. But let me reassure you that just everything I showed you with the spinal cord cells, we can do with the hippocampal adult neurons as well, okay, in our group, okay? Steve Lambert, who's really a card-carrying neuroscientist, and Amal Bahal, who's an engineer. This is the faculty of the Hybrid Systems Lab. This is the staff and postdocs. The collaborators, Sadipta Seal has made the cerium oxide nanoparticles. I've been a very good collaborator. Mark Kinney did most of the in vivo experiments that we're doing. And then just to point out, so Kerry Wilson did most of the myotube on cantilever work. Minak Das is here with his lovely wife, and a brilliant cell biologist. And just finishing up his PhD, did all the neuromuscular junction, all the adult work. I'd like to thank them. I'd like to thank the organizers for inviting me, and also thank you for your attention.